All right, let's talk about some tricky concepts related to Chapter 15, the digestive system. And I think a great way to start is looking at the specifics behind the digestion of each of the three primary foodstuffs: carbohydrates, proteins, and fats. This will bring in a lot of other concepts into play, including enzymes, sites of action for these enzymes, sites of absorption, absorbable units of these foodstuffs. So let's take a look, first of all, at carbohydrate digestion. And there's a nice summary table. It's table 15-5 out of the textbook, page 466, if you want to look at a summary of these processes. But carbohydrate digestion, the goal of carbohydrate digestion is to get polysaccharides, these strings of sugar molecules, broken down into monosaccharides, single sugar molecules. Those monosaccharides are the absorbable form of carbohydrates. So where does it all start? Well, carbohydrate digestion starts in the mouth, thanks to the secretion of an enzyme called salivary amylase. And salivary amylase initiates the digestion of these polysaccharides down into disaccharides. These long strings of sugars are broken up into smaller chains of two sugar molecules. And that's the end of sugar digestion, carbohydrate digestion in the mouth. When this food is partially digested, food is swallowed into the stomach, there may be a little bit more digestion of the carbohydrates that continues there, but essentially it's stopped because the pH of the stomach is so low, only about 2, that it effectively destroys enzymes like salivary amylase. However, when this partially digested food, called chyme, leaves the stomach and enters the small intestine, we resume carbohydrate digestion. There in the small intestine, the yet-to-be digested polysaccharides are exposed to another amylase. This time it's pancreatic amylase. The same enzyme, this time produced and secreted by the exocrine pancreas, secreted into the small intestine. So the remaining polysaccharides are broken down into disaccharides in the small intestine. To finish off the digestion of carbohydrates, to take these disaccharides down to monosaccharides, we have to utilize enzymes that are expressed on the epithelial cells of the small intestine itself, that is, on the brush border. The small intestine and the cells of the small epithelial cells of the small intestine express these enzymes on the surface of their cells so that they're continuously in contact with material that's going through the lumen of the small intestine. These enzymes finish the digestion of carbohydrates, resulting in monosaccharides, single sugar molecules that are easily absorbed in the small intestine. All right, let's now move to protein digestion. And that's going to take us into the stomach. And that's going to take us to a nice summary figure, or table, I guess the book calls it table 15-2. There's a common misconception that protein digestion happens in the stomach. It's initiated in the stomach, but it certainly isn't finished there. It's an important step. The processes in the stomach is an important first step in initiating protein digestion. So let's focus on the stomach. And when we do so, we're going to focus on 3 important cell types, and I'm going to draw them schematically on the board here. Those 3 squares are representing 3 functionally important cell types involved in the initiation of protein digestion in the stomach. The first cell type I want to draw is chief cell. And the chief cells secrete into the stomach, they secrete pepsinogen. Pepsinogen is an inactive enzyme. The other cells that are going to be responsible for acting that enzyme are the parietal cells. Because the parietal cells produce hydrochloric acid, gastric acid. Actually, in more detail than the book presents, but I think an important detail, the parietal cells secrete individually hydrogen ions and chloride ions. That is, they don't store hydrochloric acid. Instead, they secrete individually these two ions, the hydrogen ion and the chloride ion. And of course, together, these form hydrochloric acid. This very strong acid quickly reduces the pH in this immediate environment around these cells down to as low as 2. This pH is sufficient to cleave pepsinogen into pepsin. Pepsin is an active enzyme. The role of pepsin is to break down proteins into large peptide fragments. So back up a second and remember what proteins are. Proteins are large strings of amino acids. And we call strings of amino acids peptides. And so much of these proteins that we ingest are extremely long with regard to their amino acid strings. Pepsin takes proteins and breaks them up into smaller units. The absorbable form of protein is the individual amino acid, and we're a long ways from that yet. Again, pepsin will break up proteins into peptide fragments, still too large for absorption, but the first step. Important first step. And that really is the end of the stomach with regard to protein digestion. It's the digestion of proteins into large peptide fragments through the acid-dependent, hydrochloric acid-dependent activation of the enzyme pepsin. Let's finish the protein digestion story, though. And that'll take us back to that table that I mentioned before. Table 15-5. From the stomach, this chyme, partially digested food, moves its way into the small intestine. As that food moves in there, of course, so come now large peptide fragments, not ready for absorption yet, but ready to be worked upon by other enzymes. So what other enzymes are we talking about? We're talking about enzymes that are secreted from the pancreas again, the exocrine pancreas. These are the enzymes trypsin, chymotrypsin, and carboxypeptidase. These enzymes from the pancreas break down those large peptide fragments into smaller peptide fragments. We still haven't achieved the goal of getting these proteins down to individual amino acids yet. That final step, the breakdown of peptide fragments into amino acids, individual amino acids, is carried out by small intestine brush border enzymes again, called aminopeptidases. So the final step is dependent again on enzymes that the epithelial cells of the small intestine possess. Now, in the amino acid, the individual amino acid form, absorption can occur. Oh, a side note. As that chyme moves from the stomach into the small intestine, a lot of acid comes with it. And that small intestine is quickly acidified. And so the pH of that small intestine becomes quite low. In fact, it becomes very, very low and too low for those brush border enzymes to function. Thankfully, secretin acts to stimulate sodium bicarbonate release from the exocrine pancreas. And this sodium bicarbonate helps neutralize the small intestine, pH balance the small intestine, which is critical for these enzymes to function properly. All right, lastly, let's turn our attention to the digestion and absorption of fats. Our fats, dietary fats, come in the form of triglycerides. And triglycerides are a glycerol molecule and 3 fatty acids. Schematically, often represented that way. The glycerol molecule and the tails of the 3 fatty acids. Now, the digestion and absorption of fats is kind of an interesting story. Essentially, no fat digestion occurs in the stomach. Now, there's some debate in books, and some books will say there is a little bit of gastric lipase action. The vast majority of digestion of fats occurs, again, in the small intestine. And there in the small intestine, a couple things happen. All you really need to have is the enzyme lipase secreted from the pancreas, pancreatic lipase. However, this whole digestion of fats is aided by the secretion of bile from the liver. Bile acts to disperse these fat molecules, these triglyceride droplets into smaller fat droplets. See, it's an aqueous environment in the small intestine, and the fats come along being so hydrophobic that they'll all clump together, maintain their hydrophobicity as kind of a big, globule of fat. And bile inserts itself into these large droplets of fat and disperses them into many smaller droplets, a process that's called emulsification. Now, really, no chemical digestion has taken place because the emulsification, this production of smaller fat droplets, hasn't changed the chemical structure of those triglycerides yet. What emulsification has done in producing many small fat droplets, it's increased the surface area upon which the pancreatic enzyme lipase can act. And so bile is said to aid or help in the digestion of fats. All right, that's emulsification and the role of bile. So let's assume we're working with these smaller fat droplets now. What's the role of lipase? Well, pancreatic lipase takes these triglycerides and breaks them down into monoglycerides and free fatty acids. And a great summary diagram of this is on page 470, and it's figure 15-23. So this enzyme lipase catalyzes the breakdown of these triglycerides into monoglycerides and free fatty acids. These constituents of triglycerides are packaged together along with bile salts into structures that are called micelles. These micelles move across the plasma membrane of the intestinal epithelial cells quite easily. So the micelles enter the epithelial cells, the first part of absorption. We're not in the bloodstream yet, but we've moved out of the lumen of the GI tract into the intestinal epithelial cells with these micelles, these absorbable little structures that have monoglyceride and free fatty acids within them. And then a very interesting thing happens in the epithelial cells of the small intestine. In those cells, triglycerides are reformed. That is, the individual monoglycerides and free fatty acids reform into triglycerides. So the whole goal, really, of breaking down the triglycerides into monoglycerides and free fatty acids was to just get them in this absorbable little package in micelles. Because, again, in the intestinal epithelial cell, triglycerides are reformed. This is really interesting. So inside those cells are now triglycerides, structurally the same as we consumed in the first place. The goal, is, of course, is to get these triglycerides into the circulation to complete absorption. How do we do that? In these intestinal epithelial cells, these triglycerides are packaged in a lipoprotein structure made from the Golgi apparatus, and this structure is called a chylomicron. This chylomicron is secreted or extruded, as the book likes to say, from these epithelial cells and enters the lymphatic circulation, not the plasma, not the blood circulatory system, but instead enters the lymphatic system first. So these repackaged, restructured, rebuilt triglycerides, now packaged in chylomicrons, first are absorbed into the lymphatic system. And of course, the lymphatic vessels will ultimately drain their fluid, lymph, back into the venous circulation. In fact, if you look at lymph at any given time, especially after a meal, you'll find lymph to be very fatty because, again, this is the initial site of absorption. So, kind of a funny twist on digestion and absorption when it comes to triglycerides. So I hope this little video helps clear up some of the trickier points regarding to digestion of the 3 food stuffs.